Welcome to lecture 14. So, here discussing about the yield simulation, how we can calculate the yield from this the last lecture continuations as a biomass. We now discussed about the biomass calculation. So, you can predict the biomass throughout the growth stages of the crop, the above ground biomass and the below ground biomass. Below ground the root biomass, above ground the canopy biomass. So, now we will be discussing how canopy biomass is influenced by the several weather parameters, and from canopy biomass how you can go for the yield simulations. Let us see the crop yield simulation. So, as you say it is a depends upon the time integration of biomass accumulation. See if we get a healthier crop, means the yield parts or the reproductive parts that comes later stage. When the crop starts growing, the initially the seedling stage or starts the vegetative stage, peak vegetative stage, then there is a flower appearance, then there is a flower grain development and the grain maturity. So, starting from the slow vegetative, peak vegetative, then flowering, grain development. So, to have a good grain yield, grain is the ultimate the economic output. To have a good grain yield, there should be a proper growth of the crop, the good biomass. The biomass is good, biomass accumulation is good, then you can have a good the economic output, the grain. So, to have a good output, we must that is you must take care of the crop at the beginning, the initial stage, so that you can get a good biomass. If you have good biomass, you can have a good harvest, economic output can be harvested well. So, that is the best that is why I say in case of the crops, we do not keep our management towards the end, do not think that the economic part comes much later stage. So, we will keep all our input management to the later stage, no, because the economic part depends upon the vegetative parts. If your vegetative growth is well, then you can have a good economic output. To have a good vegetative growth, good biomass, you need to take care of the crop at the beginning, very beginning, from the nursery, from the initial seedling stage, the initial stages there should be a good management, optimum management of water and nutrients, and the plant should not suffer from pest and diseases. Then you can get a good biomass, good growth of the crop. Once you have a good growth, then you can have a better economic output. So, this the environmental driver control the yield through the affect the environmental parameters. They influence the dry matter production as we have discussed very detailed, particularly the effect of the radiation and the effect of temperature. So, radiation that influence the very direct influence on the dry matter productions and the leaf rate development, and the temperature on the crop development rate. So, both either the radiation, temperature, of course, the soil should be well supplied with the water and nutrients, and in that the crop is not suffering from any pest and diseases, then you can get a good biomass, and ultimately the biomass is converted to the grain yield. So, this is how the we have come to in the earlier last class, we have come to this stage only how we can get the biomass. So, now we will go into how the biomass can be converted to the grain yield formation in this simulation model. So, before that very briefly, so if you go for the dry wet, how they are influenced by the different weather parameters, the effect of temperature on dry wet, varying temperature will have the variable effect on dry wet definitely. So, you have the low temperature to very high temperature. With the increasing temperature, the dry wet of the crop increases with the days, the dry wet with crops with the gram per meter square with the different temperature range. If the day temperature and night temperature is the 18 degree day temperature, 8 degree night temperature, or 22 degree 12 degree, 38 degree 28 degree, 30 degree 20 degree. So, this graph shows that if there is a rising temperature, definitely there is a increasing dry wet accumulations, but not with the very high temperature. If you go 38 28, so and it does not give a higher yield, rather the 30 20, the temperature at 30 degree daytime temperature and the 20 degree night time temperature, that gives a better higher dry wet as compared to 38 28, because we need just optimum temperature. The range of optimum temperature, the enzymatic activity is much higher, the growth rate is also higher. So, that indicate the higher biomass production. This is how the temperature, how the temperature influence the dry matter productions. And similarly, if you see the at particular temperature, how the light intensity that influence the dry matter productions. If temperature we keep 30-20, that gives the maximum dry wet productions. At 30° day temperature and 20° Celsius of night temperature, we get the maximum dry matter productions. So, with that the see higher light flux, if the light flux increases, there is a increasing biomass. Optimum temperature, there is a increasing light density, light flux, you get a increasing the biomass production. So, that is what say of course, with the temperature remains optimum, of course, the other parameter definitely remains optimum, all management there is optimum. Then we can have if the crop dry weight is influenced by the the daytime with the by different light intensity. With the increasing light intensity, there will be increasing biomass productions with the optimum temperature range. So, yield responds strongly to available radiation under potential conditions. If the temperature is optimum, all other conditions are optimum, then the yield is strongly correlated to the radiations. So, light availability is primary driver of yield when water and nutrients are non-limiting. So, definitely say, so the crop does not suffer from any type of the stress, nutrient or water stress. Of course, there is a no disease and the pest infestations. See, with the increasing light intensity, so there will be the increasing in the not exactly you can go on very high light intensity, that has also may have the, because it depends upon the enzymatic activity at optimum light, optimum temperature. And the see leaf area development, leaf area index also, the how the leaf area index also with the yield formations, light interceptions at the different temperatures, there is a with the increasing temperature, there is increasing the leaf area enzymatic activity increases, but definitely the not very linearly. So, with the optimum temperature, with the good optimum temperature, there is a higher temperature promotes definitely faster leaf area index, but faster canopy closure increases cumulative photosynthesis. Definitely the LI dynamics indirectly regulate the final yield. Temperature same also like light. So, there is optimum temperature, we go to optimum temperature range. So, there is a higher leaf area index. So, the excessive temperature, high temperature, that increases the respiration rate, there is a more and loss of carbohydrate, and also high temperature that does not very favorable for this enzymatic growth. So, there are many enzymatic activity in the plant processes. So, the activities are optimum at the optimum temperatures. So, with increase, if you go to up to 25°C, say there is a definitely rise, say average temperature, see the increase 10, 15, 20, 25. So, there is a increasing leaf area index, but we go for the again further rise, that may not increase the leaf area index. So, this is how the this influenced by the weather parameters, and they influence the crop growth and development process. And we have discussed up to this now the canopy dry weight, how the canopy dry weight is is influenced by these weather parameters. We have calculated canopy dry weight, and see the time versus dry weight is the S-shaped crop growth curve. So, the dry weight increases with time, as the flowering comes, then the curve flattens. So, there is a particularly there is no increase in biomass after flowering, that remains almost same. Then maturity, then the crop there is a decline in the biomass. But here after flowering, what happens, there is a grain filling starts. So, though there is no increase in biomass. So, there is a the biomass, the stem weight goes on decreasing, leaf weight goes on decreasing, but grain weight goes on increasing. So, since the grain filling start after flowering, spikelet formation, grain filling starts. So, there is a partitioning, as discussing how partitioning takes place. So, here the after flowering, the partitioning within the canopy parts, either the partition in the biomass to the stem portion, leaf portions and the root portions. The before flowering, more biomass goes to probably leaf and stem the leaf portion, then some cases comes to stem portion, but after flowering more biomass goes to grain portions, the grain filling starts, grain weight goes on increasing and the stem weight goes on decreasing. So, also the leaf weight goes on decreasing. So, this is what the partitioning in the above ground the canopy parts, how much dry matter to be partitioned to the grain portion, how much to the stem portions, how much to the leaf portions. So, here as you say from here if you know the partitioning among the canopy, the partitioning the biomass to the leaf, stem and the grain, then we can also predict the grain yield. So, we will discuss this one with the particular in a software. So, here what happens after the this biomass assimilates, it is converted to dry weight growth rate, then you go for the canopy dry weight, root dry weight. Of course, here the light interception is there. Also, we are discussing about the earlier we have discussed about radiation interception, how the biomass, particularly you have calculated the biomass is equal to radiation use efficiency into radiation interception. Radiation efficiency gram per megajoules, this is gram per megajoule, this is megajoule per meter square per day. So, that gives your the gram per meter square per day. So, as the growth rate to calculate the gram per meter square per hour per day, we can calculate this one if you know radiation use efficiency and the radiation interception. Radiation interception, this also that changes over time. So, you can see the this will lead to the biomass, where it is the parameters are influencing here. If you see radiation interception, that can give to the dry weight growth rate. So, the dry weight growth rate is the radiation interception. It can be converted to dry weight growth rate using this RAE, radiation use efficiency, and this radiation interception, there are many variables they influence radiation interception, that is the leaf area index LAI, K value and also light. So, these are the variables, this is the variables, this is the variable, is a variables, this is the parameters, this is the coefficient, this is the light parameters. So, they influence the radiation interception, the state variables, and that can be converted to dry weight growth rate using this radiation use efficiency. If you like your the conversion efficiency, similarly can use radiation use efficiency, that can be converted to dry weight growth rate. And after the dry weight growth rate, then comes partitioning to the root portions, or you can get the then you can get the canopy weight, or we can get this root weight, root canopy, then partitioning, and again from canopy the partitioning to the leaf, the stem and to the grain. Finally, from this canopy we will get the grain yield. So, how this is done? If you see, I can give example of a crop, you see the rice crop. So, now we have come to this stage canopy dry wet. How the canopy dry wet can be partitioned to leaf, stem and grain like this way. After the flowering, we know they have got the canopy dry wet with different by calculation from this using the radiation interceptions, using the crop parameters, leaf area, using the light extinction coefficients, and we now we have got this canopy dry matter. Now, the point comes how the canopy dry matter will convert it to the grain yield. The grain is the economic parts, how the canopy biomass will be converted to the grain parts. So, let us give an example of rice crop. If you see rice crop, this is the soil portion, the underground soil, the root portion, then these are the leaf, portion, and you get the grain here. So, this portion is the grain portion, we say panicle, and this individual grains. Hold the panicle, individual grains. When you go for the how the conversion takes place after the this flowering stage, when crop goes to flowering. So, there is a grain formation and there is partitioning. After flowering, partitioning, stem weight, leaf weight and grain weight. So, there is a the partitioning to stem weight goes on decreases, leaf weight decreases and the grain weight partitioning increases, that is a grain filling stage. Come to this point, this is grain filling stage. We in case of the crop model this chart will be discussing how the model works, they are partitioning. At flowering there is a parameter is there, genotype parameters. One is G1, that is number of spikelets, that talks about number of spikelets per gram of culm at flowering. So, the culm means this is the culm portion, you can say this portion, the above ground portion, this is the culm, no leaf blade, excluding leaf blade, this is the culm portion. So, the G1, this parameter, number of spikelets per gram of culm weight at flowering, per gram of culm weight at flowering. So, this is the what is spikelets? These are the individual spikelets. So, after filling that develop into the grain portion. So, the number of spikelets per individual culm weight at flowering. So, at the flowering stage, so with the culm weight, so for that culm weight, because we calculate the dry weight, because from the canopy dry mass we get the partitioning, we get the culm weight, stem weight, and also we get from particular stem weight how many spikelets will be formed. So, that is the partitioning coefficient per particular 1 gram of the stem weight, how many spikelets will be formed, that talks about the partitioning of the canopy dry matter to the grain portion. So, this is give G1. So, you have G2, G2 talks about the single grain weight, gram. So, if during partitioning, if we know the canopy dry weight and the fraction that goes to culm, if you know the culm dry weight and this particular column dry weight, how many grains are there? This is for the how many grains are there. Suppose you say G 1 in case 60, the value comes G 1 is equal to 65. That means 65 number of grains, 65 grain, it indicates 65 spikelets. Each spikelet that will be converted to a grain at maturity grain, 65 grains per gram of column weight. So, during partitioning, so that will give the partitioning also the fraction goes to column. So, if the whatever the column weight comes, if the column weight is simulated, then we can simulate how many grains are there at the time of maturity, how many grains will be there in that rice crop. In case you see, suppose we simulate the 1 gram or you can say per 1 meters. If you see, in case you see, suppose for a particular area we get, for example, we get the column weight with the simulation process, we get the column weight, column means this is the column weight 100 gram per meter square. So, if 100 gram per meter square is the column weight, so this conversion is 65 grains per gram of column weight. So, if you multiply 65 grains per gram of column weight, so 100 gram per meter square, 65 grain per gram. So, that gives the grain number we get 66,500 grains per meter square. So, per meter square, using this simulation we can get 6,500 grains per meter square. And the second component, single grain weight. The single grain weight is a genetic parameter of this crop. This hardly varies with your management practices, that remains almost you can say similar. So, if you single grain weight in genetic is a constant, we can give suppose G 2, if the G 2 value is known, suppose 0.02 gram G 2, if G 2 0.02 gram. So, the grain weight we can calculate the grain weight is equal to the now we have got the based upon the column weight, we have got how many 66,500 grains per meter square. So, the grain yield from here we can know grain yield is equal to 6500 grains per meter square into each grain weight 0.02 gram. So, that gives 6502 into 0.02, that becomes 130 कितना आ रहा है 130 grams per meter square. So, from this we can say, we can see the grain yield comes around 130 grams per meter square, we can calculate. So, this is how the grain yield calculations, grain yield simulations using crop model, because that depends upon the biomass simulations. Once you come to biomass, then model has a factor after flowering how much we partition to the different plant parts, either the leaf weight, stem weight and grain weight. And from this partitioning we know the column weight, how much column weight exactly at the time of flowering based upon the partitioning. And if you know the column weight and then the number of spikelets per unit column weight, per gram column weight at the flowering, this is a parameter in the model based upon the experimental understanding. If you go for through rigorous experiment, so that parameter for a particular variety, this is not a same for the all varieties for a particular crop. That varies from crop to crop definitely, and in a particular crop that varies from the cultivar to cultivar, that means variety to variety. So, particular variety, this conversion of the number of grains per unit column that varies among the varieties. So, if this value is known from this for a particular calibration and validation process, if you know this value, if you know the G1 value and also G2 is a genetic parameter of the cultivar, that is a very specific cultivar, that is also known. So, multiplying G1 and G2 based upon the column weight simulation at the time of flowering, we will be able to simulate the grain yield at the time of maturity. So, the point is that the biomass that can be simulated up to flowering stage, most of the cereal crops you can see, from that biomass you can predict the grain yield to be at maturity, it takes around 25-30 days maturity. Based upon these parameters, the conversions of you can say G1 is the conversion factor from the column weight to the grain number. So, how much grain number can be for a particular variety depends upon the column weight at the flowering. So, based upon the column weight of the flowering, if you simulate get the column weight at the flowering stage, from the column weight we can calculate number of grains can be achieved, can be attained at the maturity. If number of grains, the G2 is a, we can say the genetic coefficients of the crop, if G2 multiplied, we can get the grain yield of the particular variety, how much grain yield is expected towards maturity. So, this is how the conversions of the partitioning of the biomass and the again biomass partitioning to the leaf weight, stem weight and the grain weight. From that you can calculate the grain yield of a specific crop. But we will be discussing detail in no particular, we will go for the rice crop and go for the some other crops also, and where is a very specific for the software decision support system for agrotechnology transfer. When you go for the explanation of software, we will be discussing how these genotype coefficients are used for the grain yield simulation when you go for the as a hands on exercise using the software. But this is a basic understanding. You have the canopy biomass, then canopy biomass is converted to your partition to the stem, leaf and the grains, and that based upon the column weight flowerings, that gives the conversion of the per unit column weight flowering, how much grains are expected, and based upon that we can calculate, can predict the grain yield of this crop. This is the what the crop yield is simulated that is as a function of the biomass accumulations over the time, because biomass is very important. So, that is why we do take care of the crops at the beginning to have a good productions, the maximize productions. The care must be taken at the beginning of the crop growth period. If you neglect at the beginning, so it is very difficult to get the output if you want to take care of the crops at the later stage. So, essentially the initial stage, seedling stage or the juvenile stage of the crop, we can say that is a very important for better management, better input management to have to maximize the productions, to have the maximize biomass and the production and grain information. Temperature, that also controls the growth rate, development speed and the leaf area expansions. Light controls the photosynthesis and the total dry matter productions. The simulation results demonstrate how environmental drivers shape the yield potential when all the managements are kept at the optimum, like the no water or nutrient stress and the crop does not suffer from any type of pest and diseases. Of course, we have discussed here the how the biomass is simulated, and from the biomass how you can go for the partitioning to the yield, grain yield. So, that depends upon the biomass at the flowering stage. If you know biomass at flowering stage, then the partitioning from the column weight, like example of rice crop column weight for a particular for a 1 gram of column weight, how much spikelets formed and the spikelet gives the grains and the single grain weight multiplied, we can get the grain, that means grain yield. That means the biomass at the flowering stage can give informations how much yield can be predicted, how much predicts the simulation of the yield at the crop maturity. So, this is how I hope it should be clear for you the potential, the use of the potential production simulation models for the biomass predictions, and from biomass how you can go for the yield prediction of the crop. With this, I thank you all.